everyone, welcome to my channel Competitive Botany. In this video you are going to learn competitive bits from plasma membrane. To get my next video and notifications, please subscribe the channel and share this video to your friends who are preparing for competitive exams. Let's go to the class. Here you can see the picture of plasma membrane. The cytoplasm of both plants and animal cells is surrounded by a membrane called plasma membrane. This is also called as cell membrane or plasma lemma. Generally, the plasma membrane may be defined as a thin, elastic, semi-permeable living membrane that serves as a boundary for the cytoplasm. Now coming to the competitive bits from the plasma membrane. Let's see the first question. The name cell membrane given by? Answer is Nageli and Kramer. Next question. The term plasma membrane given by? Answer is D. Robertson. Let's go to the next question. Cell membrane is composed of lipids and proteins. Actually, the cell membrane is mainly constituted by lipids, proteins and also carbohydrates. But here the carbohydrates are in least proportion. Now coming to the next question. In plasma membrane, structural and functional role is played by? Answer is proteins. Answer is proteins. Proteins constitute 70% of the plasma membrane. Let's go to the next question. The main work of cell membrane is it controls the entry or exit of substances in the cell. Next question. Now it is time to discuss about different models proposed by different scientists to explain the structure of plasma membrane. Let's see the first model, that is Lipid Bilayer Model. This bilayer model was proposed by two scientists. They are Gorter and Grendal. Here you can see the picture. According to them, the plasma membrane contains lipid bilayer. You can see the two layers of lipid molecules arranged in this model. Okay, so they also concluded that polar groups of lipid molecules were directed towards the outside of the bilayer. So you can see hydrophilic heads are facing outside. You can see the picture. Okay, so now going to the next slide. So before going to the next model, you have to know one thing, that is regarding lipids of plasma membrane, the structure of lipids. So cell membranes are mainly composed of lipoproteins. As I told you before, the most common lipids found in the membrane are phospholipids, glycolipids and sterol. All of them are amphipathic. That means they have both hydrophilic and hydrophobic portions. So you can see the picture. So this is the head part and this is the tail part. Okay, so in this picture you can find that hydrophilic head and hydrophobic tail. Okay, so that's why these lipid molecules are in amphipathic in nature. They are in amphipathic in nature. Okay, so now going to deal the second model. This is called as sandwich model. Sandwich model was proposed by Danelli and Davson in the year 1935. You can see the picture. This is called sandwich model. This model is also called as bimolecular leaflet model. Bimolecular leaflet model. This model states that the same points as mentioned in the above model, but the only difference is the lipid bilayer sandwich between two continuous layers of globular proteins. You can see the globular proteins in the picture. Okay, so that is only the difference among bilayer model and sandwich model. Next model, unit membrane model. This is the third model proposed by the scientist Robertson. He found that all biological membranes appear to be made up of lipid bilayers. They have the thickness of 35 angstroms and a dense band of proteins either side. They are having 20 angstroms thickness present on either side of the lipid bilayer. So this model is also called as trilaminar concept. This is also called as trilaminar concept. You can see the picture. So lipid bilayer is having two types of proteins. They are present either side of the lipid bilayer. This model is also called as trilaminar concept. It is also similar to Danelli and Davson model, except that the protein layer at the exterior and interior are different. The proteins present exterior and interior are different. Okay, so next unit membrane model was proposed by the scientist Robertson. Coming to the next model, that is the important model, is nothing but fluid mosaic model. Fluid mosaic model was proposed by the famous scientists Singer and Nicolson. According to them, the lipid molecules are present in a fluid state capable of rotating and moving laterally within the membrane. The protein occur discontinuous particles that penetrate deeply into the lipid sheet. The protein particles resembles floating icebergs in the sea of lipids. You can see the picture. So this was this model, fluid mosaic model. This is the famous model, widely accepted model proposed by the two scientists Singer and Nicolson. There is another model too, that is fifth model called as Micellar Model. According to them, plasma membrane contain repeated globular subunits known as micelles. You can see the picture. So plasma membrane is made up of these repeated globular structures. So this model was proposed by Hilleir and Hoffman. These are the models are proposed to explain the structure of plasma membrane. Now coming to the next question. Which model is accepted and why? Fluid mosaic model was accepted model because it explains the osmoregulatory process. Next question. In the fluid mosaic membrane model, the phospholipid bilayer has proteins embedded in it. Next, coming to the next question. Differential permeability of plasma membrane is explained by, answer is fluid mosaic model. Coming to the next question. So according to fluid mosaic model, plasma membrane is composed of phospholipids, extrinsic proteins and also intrinsic proteins. Now coming to the next question. Singer and Nicolson model of plasma membrane differs from Robertson model in the arrangement of proteins. It differs from the Robertson's model in the arrangement of proteins. Next question. In fluid mosaic model, highly viscous and impermeable to water soluble molecules are, answer is phospholipids. Answer is phospholipids. Next, coming to next question. According to the fluid mosaic model, the extrinsic and intrinsic proteins are arranged in a sea of lipids. Answer is asymmetrically. Answer is asymmetrically. Here you have to know one thing, that is fluid mosaic model is applicable to both prokaryotes and eukaryotes. It is applicable to both prokaryotes and eukaryotes. Next question. The most abundant lipid in cell membrane is, the answer is phospholipid. That means phospholipid is an important constituent of cell membrane. Now coming to the next question. Plasma membrane is freely permeable to solvent. Plasma membrane is freely permeable to solvent. Next question. Cell membrane is concerned with active uptake of ions. It is concerned with active uptake of ions. Next question. Osmoregulatory process of cells is under the control of plasma membrane and vacuole. Osmoregulatory process of cells is under the control of plasma membrane and also vacuole. Next question. The movement of water molecules from the less concentration hypotonic solution of the medium to the inside of the cell is called endosmosis. Next question. Plasma membrane is clearly visible in plasmolysed cell. To observe the plasma membrane clearly, the cells must be plasmolysed. Next question. Glycoproteins and glycolipids are important for cell-to-cell recognition. Cell-to-cell recognition achieved by membrane carbohydrates. Cell membrane is mainly constituted by lipids, proteins and carbohydrates. As I told you before, carbohydrates are in least proportion. They are important for cell-to-cell recognition. You should remember. Next question. The membrane proteins never been observed flip-flop across the membrane because they are restricted by the carbohydrates. Next question. The side-to-side movement of the phospholipid is known as the side-to-side movement of phospholipid is known as intracellular diffusion. Now coming to the next question. Lipid nature of plasma membrane is destroyed by, the answer is benzene. Now coming to the next question. Name the specialized regions of cell membrane useful for addition of cells in animal tissues. The answer is desmosomes. Coming to the next question. Each gap junction is made up of specialized protein channels called connexins. Before going to leave this question, you know what are gap junctions? Channel proteins of opposite cells. You can remember this. Okay, so that means proteins that are present in the plasma membrane, those are the channels for the transport of molecules inside and outside. Okay, so gap junctions nothing but channel proteins of opposite cells joined together, providing a channel for the transport of ions, sugars, amino acids and other molecules to pass. They allows communication between cells. Example, gap junctions present in heart muscles and also in animal embryos. Okay, so next question. Here I have to tell you one thing also. Each gap junction is made up of specialized protein channels called connexins. called connexins. Okay, so next question is, urine does not seep out because of tight junction. Okay, because of tight junction. Now coming to the next question. Plasmodesmata in plant cells similar in function to gap junction. Okay, next question. The proton pump in plant cells is the functional equivalent of animal cells. Answer is sodium potassium pump. Answer is sodium potassium pump. Next question. Porins present a large family of proteins that form pores on the membrane by beta barrels. Answer is beta barrels. So I will make the next video on the role of proteins in membrane transport and types of transport proteins. So please watch my next video too. Okay. Now coming to the next question. In a prokaryotic cell, the respiratory enzymes are present in. Answer is plasma membrane. So mesosomes are the infoldings of plasma membrane. They contain respiratory enzymes. Thanks for watching.